A-Level Biology 9700, Paper 5, Period 3, May, June 2024, question number 2. And question number 2 is, uh, yeah, gibberellins are a group of plant hormones that are involved in the elongation of plant stems. A student investigated the effect of two different concentrations of gibberellin known as GA3 on stem elongation of 10-day-old pea seedlings. The student was given a stock solution of GA3 with a concentration of 2.85 into 10 raised to power minus 4. Describe a method the student could use to make a solution of GA3 with a concentration of 1.90 into 10 raised to power minus 6 and state the dilution factor used. So first of all, we will try to find out the dilution factor. Then we will describe the method. So first, the formula to calculate the dilution factor is, that is, we will divide the concentration concentration of stock solution uh, with the solution which we want to make. So we will divide 2.85 into 10 raised to power minus 4. We will divide it with 1.90 into 10 raised to power minus 6. So after calculating, I'm just calculating right now. After calculation, we will find out that is 2.85, 1.90. So the dilution factor is equal to, factor is 150. So we will take 1 cubic centimeter of stock solution and will add it in 149 cubic centimeter of distilled water. And the next question is, first of all, dilution factor is necessary, then you will be able to find out the exact uh, amount of the stock solution to be added into the distilled water. So in the investigation, the student used the two concentrations of GA3, 2.85 into 10 raised to power 4, high concentration GA3, and 1.90 into 10 raised to power minus 6, low concentration of GA3. Applied the high concentration GA3 to one batch of 10-day-old pea seedlings. Applied the low concentration GA3 to another batch of 10-day-old pea seedlings. So both concentrations high and low. Then applied distilled water to a third batch of 10-day-old pea seedlings as a control. Standardized all the variables. Measured the length of the stem of each seedling every 2 days until the seedlings were 20 days old. So calculated the rate of stem elongation in centimeter day 1. So state one way in which the student would standardize the measuring of stem length. So he want to standardize the measuring of the stem. So for this purpose, uh, I think he can measure in appropriate units. So we can say that there should be the appropriate units in which it should be measured. Secondly, the stem should be in a straight position, and the measuring tool should also be in the straight position. So, in this way, you can say that the measurement can be standardized, or a measuring tool should be appropriate. Measuring tape, you can say it is not an appropriate method to measure, but we can use it. So here I am going to write the points which should be the only for one point. So first of all, I think it is the stem should be to get accurate value, or measuring tool can be used. Now the next question is, the results of the investigation are shown in table 2.1. Age of seedlings and the mean stem length with high concentration Ga3 added per centimeter, mean stem length with low concentration Ga3 added per centimeter, mean stem length with distilled water added per centimeter. So at the end, they have found the rate of stem elongation per centimeter. So complete table 2.1 by calculating the rate of stem elongation in centimeter per day for the seedling with low concentration GPA3 added. So the blank which they have uh, in the table, we have to calculate the rate of stem elongation. So for this purpose, uh, we will calculate in this way. They have the space given for this purpose. Initial, first of all, we will write in this way. The mean stem length with low concentration, which will be the final at 20th day. So we will final value minus initial value divided by days. So the final value is 21 and the initial value is 2. We will divide it. Days are 10. 10 to 20, there are 10 days. So it is 19 by 10 will be equal to 1.9. So the value is 1.9. So this is the way to find out the rate of stem elongation. Rate of stem elongation. Now, next question is, a scientist stated that they did not have enough confidence in the results in table 2.1 to make any conclusion. State one reason to support the statement made by the scientist. So, the statement which was made by the scientist, here I would like to say that he measured all the lengths in centimeter instead of millimeter, because millimeter, if we measure we take the lengths in millimeters. It is a, you can say, finer unit of measurement. Millimeter is a finer unit of measurement, allowing more precise control over the dimensions of the components or the lengths which we are going to measure. So millimeters are the standard unit of measurement in many cases. So millimeter is more precise unit. So we will write this. This I have told you, that is length was measured in, I will add instead of millimeter. So we measured in mid centimeter instead of millimeter, and no statistical test was used for to calculate. So next question is, describe how the student could modify the investigation to increase the confidence in the results. As I have told you, the millimeter is more precise unit in, so, or standardized unit. So we will write the measurement should be in millimeters, or more samples can be used for this purpose. Or the number of samples can be increased. Next question is, the student found a study on the internet that considered the effect of different wavelengths of light on the concentration of two types of gibberellin GA1 and GA8 in pea plants. In the study, 60 pea seeds were germinated and kept in the dark. For 7 days, the pea seedlings were divided into 4 batches of 15. Batch 1 remained in the dark. The batch 2 was exposed to blue light, 470 nanometer. Batch 3 was exposed to red light, 680 nanometer. Batch 4 was exposed to far red light, that was 750 nanometer. After 4 hours, the concentrations of GA1 and GA8 in the seedlings were measured. So the table 2.2 shows the results, and here you can see the wavelength has been given, the color of the light has been given, the mean GA1 concentration, and standardized deviation. Standard deviation per fresh mass has been given in the plus or minus values. On the other hand, the mean GA8 concentration plus and standard deviation over fresh mass has been given. These 4 columns has been given. The student calculated the standard error and 95% confidence intervals for the data shown in table 2.2. So the data has been given over here. The formula that the student used were SC is equal to S over under root N is equal to 95% calorie is equal to mean value plus minus 2 into standard deviation. So keys to symbol they have given us now. In the next table, 2.3 shows the calculated value for SE and 95% CL for the data from table 2.2. So we have to find the values for column 1 and column 2. So complete table 2.3 to show the calculated value for SE and mean plus minus 95% CL for the data from far red light for GA G1. So here you can see that the standard deviation is 0.26 in the column 4. So the column 4. So we have to find out with the by keeping this standard deviation value 0.026, which is this one. We will make use of this value 0.26, and then under root, and there are sample size is 15. So you can see that 15 plants were used in each batch. So the sample size for each batch was 15. So we will use this formula and the values, and we will this formula and the value 0.26, which has been given in the table 2.2. We will make use of these values and we will calculate the first of all SE for G1 and mean plus minus 95% CL for GA1. So here I would use this formula is equal to SE over N, which has been given over here. So it will be S over, and it is equal to SC. We have to, so when 0.26 over 15, so 0.26 over 3.87. So the answer you can also calculate with me, so you will better understand the way I have calculated. So it is 0.26 divided by 3.87. It is equal to 0.067. So we will write 0.067. Now we have to find out the mean plus minus 95% CL. So 95% CL is equal to mean plus minus. So for this purpose, we will, it is equal, we will take the formula from the back as usual. Mean is equal to this formula. Now we will use this formula. And the mean is, we will take the mean value. This is the mean from the table 2.1, which is 1.38. So here we will write in this way. So it will be 1.38, which has been given in the table 2. Plus minus, we will multiply 2 into 0.067. So it will be equal to 1.38 plus minus 0.13. You can also calculate in this way. So I have calculated in front of you. You can also calculate by yourself. So you have to you have to take the values from 2.2 and then you have to calculate. Now here, next question is 2.1. Figure 2.1 shows the bar chart of the results from the study, and 5, 4, 3, 2, 1. So the mean Libralian concentrations are given on the y-axis and 4 batches are given on the x-axis. So they have shaded the GA1, A8 and GA1 is plain without any shade. So use figure 2.1, table 2.2 and table 2.3 to state 3 conclusions about the effect of different wavelengths of light on the concentrations of GA1 and GA8 in the plants. So from these tables, uh, 2.2 and table, this figure 2. We will use this figure and we will compare the values. And here you can see from this table, you can see the highest concentration of the batch. I will also write, there were no, it was 470, it was 680, it was 750. So you can see the highest value of G8. This is the G8 which has highest value in batch 3, in which there is red light, which is 680 nanometer. And you can also see the lowest value GA1 in the batch 470, and which is blue light. And you can also see the highest value of GA1 is in the first batch where there is no light. So, and the on the other hand, we can see the we can see that the third one, 95% does not overlap. For the first concentration, you can say little bit they are different. They are different. They are not overlapping. For the lower concentration of GA1, for the all wavelengths of light, they are different. There are great difference between these two. So now we are going to write all this. I have explained. We are going to write now. So here you can see there was no light. There was the light was 470 nanometer, and there was 680 nanometer, and there was 750 nanometer. So the highest, you can see the highest concentration of GA8 in red light with 680 nanometer. And on the other hand, there is lower concentration lower concentration of GA1 in the blue light. GA1 has lower concentration in all wavelengths. You can see from the graphs. that it is the lowest in all wavelengths. Now you can also see from the table, that is, I think we have written the three answers. Beside this, we can also write that 95% E does not overlap for all the wavelengths of light. So there is a significant difference between them. So it was all about the question number two. Please like, share, and subscribe my channel to watch the videos on papers, or you can say on syllabus as well. Thanks for watching.